Mm. Hmm? You can see the slide?
Yes, we can
Okay.
see it.
Yeah. Okay. So this is the third time lecture,
Yes.
and title is Review the Old to Gain New Insight, Discoveries from Fermented Food. So we have, I will introduce some um, fermented food and new discoveries from that fermented food. Okay. So here is a list. So at first I will talk about Narezushi. This is a fermented fish. So this is in that case we use sweet fish or fragrance fish. This fish smell is like cucumber or some vegetables because they eat lot of algae in river. And this is a very famous. You mean the Yorani sensei, Professor Yorani knows well about this fish. Because Gifu is famous of this fish. And we use this kind of bird, sea cormorant, for fishing. So traditionally, about 1300 years ago, this was started. And we say cormorant fishing. And I got sweet fish. And this is, sorry, cannot see well. So anyway, here, some loop was tied loosely because they can eat small fish, but they cannot eat big fish. So big fish, we got that is the system. So if they take small fish, they can eat. So anyway, you see here, fish is catched by this bird. So did you go, Yoran Sensei, Professor Aaron, did you go for, no,
Yes, I go it. Yes, I went because Gifu University
yeah,
provide to foreign people, foreign
yeah.
student. So I join in this festival.
Okay. Cute. We have this festivals anyway in Gifu, and this is very famous. And, oh, sorry, what? What? Okay, okay. So, just a moment. Mm, Cannot see well why. Oh, okay, I could. So, this is how to make ayu narezushi. Narezushi is a fermented food mm, with fish and rice. And here we remove the organs from fish and we put the salt and salting the fish for 1 month or several months. And after that, we desalting in water and with rice. We put in the case bottle and pressing with stone and fermentations for 1 month, or it depends on the producer. And we have fermented fish mixture, and this is iron Narezushi. So this is very old or long histories about this Narezushi. Okay. And we check what kind of microbes are abundant in this iron Narezushi. So this shows a heat map of the OTU. OTU means genes where come from. So this means species, no, genus, like that. In this case, family. Anyway, in Gifu iron Narezushi, we found many lactobacillus lactic acid bacterium after fermentation. And some enterobacter are here, but this is a past, and after 13 days and after 26 days fermentation, it become better, and lactic acid bacterium become dominant. So this is a fermentation process we understand. But we don't know how this microbes come from and where it come from. So that was a question. We know many Narezushi was produced by lactic acid bacterium, but we don't know where it come from. Sometimes we say in the house that producing Narezushi, or from bottle, or sometimes or fish, and airs. We don't know where it come from. So that was the target. So how we see the origin of lactic acid bacteria? So mainly, probably we thought it came from fish, because rice, probably no, because rice is already heated, and maybe come from some these bottles, especially wood bottle can be the source. So we check the origin of lactic acid bacteria with this kind of experiment. So sweet fish and salt, this is general method, and unsalted, and sweet fish and rice, and we put that in plastic bag, not using these bottles. Okay, and we check the fermentation, and we check the families, OTUs, using NGS, next generation sequencer, and we found after 26 days, salted fish become similar diversity, because lactic acid bacterium become dominant. Maybe enterobacteria sometimes here, but without sorting, we found there is Aeromona desu, So Aeromonas related microbe. This sometimes cause disease. So we found that salt is very important to kill this kind of microbes. And with this experiment, we try to find confirm microbe come from sweet fish or not. And the plastic bag have no microbes. So this shows microbes come from fish, from feed fish. We don't know why this lactic acid bacteria came from sweet fish, but anyway, we found from fish it come. And we also found salt curing is important step to avoid non-flavored microorganisms. So probably Indonesia, you also have fermentation food using
Fish.
fish
Mm.
or meat.
Yeah, we have it.
And you also use salt.
Yeah, we also use salt too.
Yeah. So salt, we found that salt is very important step to avoid non-flavored microorganisms. But probably this is to kill microbes. Sometimes we protect the growing of microbes. We use salt. But anyway, I will talk about effect of salt again later. Anyway, what I want to say is salt curing is very important to stop, avoid non-flavored microorganisms. And lactic acid bacteria came from sweet fish. This was not confirmed. And indeed, for us, it is strange because lactic acid bacterium sometimes come from plant or environment. Especially these lactic acid bacterium case is lactobacillus is very popular indeed. So in that case, we thought that came from plant or factory, but it came um, from fish. This experiment showed, I think. Okay, this is one kind of new insight because no one proved ferment or lactic acid bacteria came from sweet fish or origin. We didn't know indeed. So okay, and next is Saba Narezushi, mackerel Narezushi. This is a Kishu Narezushi. I already introduced student that my hometown produced this. And we found unexpected organism from Saba Narezushi, of Kishu Narezushi. And that was produced Yasuke Sushi Bar. This is a name of shop. I already talked, but anyway, similar to Ayu Narezushi, Kishu Narezushi or Saba Narezushi is made like this. So salted fish, and after desalting, we make sushi homing and packed in a barrel and put weight and fermentations. So this is very similar. And this is a Narezushi. And the difference from Ayu Narezushi is we eat rice and fish together. But in the case of Narezushi, we mainly eat fish. So, and I showed you that which kind of microbes come from, which kind of microorganisms become dominant. I already talked. And if you see, maybe there is a student who already understand or find the strange result. We found bifidobacteria in fermentation. Generally, in Japan, we have no report isolation of bifidobacterium from fermentation food. We always get that from stomach, animal stomach, or baby's stomach, like
Yeah.
that.
Baby poop too.
Yeah, generally. But we found this in fermentation food. In Japan, we don't use milk for fermentations, only fish. So that is why we don't have the experience or report about bifidobacterium isolation from fermentation food. So I thought this can be the first case of isolation of bifidobacterium from fermentation food. And we tried. At first, we tried to confirm there is really bifidobacteria in this sushi. Because this is just NGS analysis. And we have to confirm that is already or exist in Kishu Narezushi. So we take DNA and PCR using bifidobacterium specific primer. Students know PCR? Okay.
They know it. They study it.
Yeah. So we found this is the electrophoresis result. And we found the DNAs, and this seems single. So we thought that there is a bifidobacterium and only one species. If there is a lot of species, this band can be different. So we tried to sequence this after getting this DNA, and we've succeeded to get primary sequence of this from this DNA. And we found this is a Bifidobacterium aquacephali, and or Bifidobacterium cycloaerophyllum. So after isolation, we can find which. But anyway, what we found is these are different samples, but only one species. So this means this microbe lives in producing house or factory or brewing or some case or anyway, origin is not from natural product. Because if this come from fish or some leaves, so it should be abundant. There must be different species. but there's only one species, it seems. So we thought this lives in house or some yeah. Anyway, environment in producing tissue Narezushi. Anyway, and sorry, same things. Brewery. We should talk here first. Anyway, they should live. They must live in brewery. And we try to isolate bifidobacteria, bacterium, not bacteria, bacterium. And this was very hard, indeed. I think we need 4 years to master course student we need. Because this is the first case. No one isolated from fermented food. We don't know, indeed, which kind of bifidobacterium and which kind of, I don't know the characterization of growth. So anyway, we try to isolate and like this, taking single colony and grown in membranous medium using the anaerobic condition. And after Sanger sequence, I try to identify. And many cases, almost all are lactic acid bacterium. But sometimes we found. And after 4 or 5 years, we could isolate bifidobacterium. And bifidobacterium was successfully isolated from Kishu Narezushi. So, and using the PCR, 16S ribosome RNA, and also HSP60, this can be the indicator for identifying which species. And we found this is a bifidobacterium cycloaerophyllum with a 99.8%. And in the case of HSP60, 99.6%. So the isolated bifidobacterium is Bifidobacterium cycloaerophyllum. This was first isolated about just 5 years ago. So if we succeed to isolate in about, well, we started the experiment first. That can be the new species, but 5 years ago, this was already identified. So it was shown that isolation has been reported from pig and some dairy product. So this came from Yasuke's shop, sushi bar, and named Bifidobacterium cycloaerophyllum Yasuke strain. And this is submitted to some culture collections. And whenever, if you need, you can get this species from that culture collection. And this is a result of scanning electron microscopy observation of Yasuke cells. Okay. So this shape, especially this, is like typical bifidobacterium. So we confirmed this is bifidobacterium. And it was very difficult to isolate, but we tried to characterize environmental stress tolerance of Yasuke strain for characterizing this species. We used Yasuke strain and DSM 22366. This is a type culture first isolated in Europe. DSM is in Germany. And as a control, this is a control, and we also compared some character with Bifidobacterium longum and Bifidobacterium adolescentis. These reported to live in human group. This is a salt tolerance. We focus on salt tolerance, pH tolerance, acid tolerance, and oxygen tolerance. So this is the growth of Yasuke strain, and DSM is here, and other bifidobacterium is this. And it seems high salt tolerance, probably because of salt treatment was employed. And this species live in brewery houses. There we produce Narezushi. And I didn't say from Ayu Narezushi, we couldn't find bifidobacterium. So that is not special case of this Narezushi. And probably only Yasuke Narezushi has bifidobacterium. Anyway, they live around in house brewery, and they have the high salt tolerance. And next is acid tolerance. We compared. You can see similar. Yasuke strain was not acid tolerant. But indeed, if you ferment in milk, Yasuke strain is resistant indeed. They survive until low pH, but this is just resistance. If you compare the growth with other species, Yasuke was most acid tolerant. We already made yogurt and cheese, but yogurt was very acidic, and probably Yasuke strain produce acetic acid, not only lactic acid, probably. So yogurt was not good taste, but in the case of cheese, it was good taste. I will not explain about that in this lecture. And surprisingly, Yasuke strain has high oxygen tolerance. Generally, bifidobacterium can grow only in anaerobic condition. But this is a static culture, anaerobic conditions, and static culture and aerobic condition. Anaerobic condition means in anaerobic chamber, we incubate it. Here, aerobic means just stand. And we shake tubes. That's Yasuke strain grows. And also DSM strain also grow very well. So probably Bifidobacterium cycloaerophyllum has high oxygen tolerance. So we don't know why we couldn't isolate so easily. if we know this, maybe we could isolate easily. But anyway, that is the finding after isolation. So until we succeed to isolate, we cannot say this is good or bad or resistant or sensitive. We didn't know. Anyway, we found um, Yasuke strain have the high oxygen tolerance, similar to type culture of DSM. So that means this strain can be used for many brewing processes. So in this case, that case, maybe natto. Natto is aerobic fermentation, but bifidobacterium can be added. This strain can be added to natto. Natto is, I already showed, and soybean fermented food used by Bacillus subtilis. So this can be used for such kind of usage. And about the draft genome for the Yasuke strain, we analyzed using NGS, and genome size was this, and the number of contig was 20. So contig means some fragment that was made. So we cannot make circular. So just a draft genome of the Yasuke strain. And we compared genetic homology and so very close to DSM strain. And we have other strains that, anyway, this is Bifidobacterium cycloalpha using draft genome sequencing and close to DSM 22366. And we also compared genome assembly and comparative genomic analysis. So we got genomic information using Yasuke and cyclophilan. Oh, this P should be small. And longam and bihidan. And annotation, and we compared genomics. And comparing genomic analysis for identification of accessory genomes of Yasuke strain. And we found, where? Just a moment. Okay. We have better see here. So what we found is spark-side dismutase and glutathione peroxide was found as a unique gene. There must be, no? No. Why? Maybe not specific, but for these strains, these strains, yeah, we share this. Anyway, but this right gene unique to the species, I couldn't see. Anyway, superoxide smooth third and glutathione peroxide Yasuke strain has. And fluorine glycine betaine we found. So this was related osmotic stress resistance. And superoxide dismutase is a enzyme to delete superoxide anion. And this is linked in glutathione peroxidase. Anyway, these enzymes contribute on reactive oxygen species dismutate. So probably this system is because of the oxygen resistance. And this enzyme or transporter was because of osmotic stress resistance. But we couldn't find the Yasuke strain of chrome genes. So this means no respiration capacity. They just decrease the reactive oxygen species, but they don't respirate. So this is our finding of this accelerated genome identification. You see proline racemase related here, but I don't know. Probably this strain has. Anyway, in this case, new insight was Bifidobacterium was successfully isolated from Kishu Narezushi. This marks the first report isolation of Bifidobacterium from traditional Japanese fermented food. So this was the first case. And the Yasuke strain exhibit high salt tolerance and oxygen tolerance. So this oxygen tolerance is very characteristic for Yasuke strain and Bifidobacterium. I forget the name. Cycloaerophyllum. And this suggests it possesses characteristics well suited to the fermentation environment Narezushi. Yes, because of high salt tolerance is Narezushi production. And high tolerance make it easy to utilize in fermentation production. So in the future, it can be used for many kind of fermentation food, even for natto. So Yasuke strain possesses unique genes associated with oxygen tolerance and salt tolerance. So, sorry, this is common to species, so not unique genes. But any specific gene, special gene. This is new insight. So probably these bacteria live in the brewer that was established about 150 years ago. I don't know when it stayed there, but only Yasuke bar we can find this kind of bifidobacterium. We are lucky. So this is next Uruka. Uruka is so sweet fish is very popular in Gifu prefecture, and we eat sweet fish, but viscera organs we don't eat. And so traditionally in Gifu area or other in Japan, we add salt and ferment anaerobic conditions, and we eat this as uruka, and we can get this kind of food. This is special. Have you ever eat, Professor Yorani? No, you didn't.
I think that Uruka, I never eat it.
Did you see?
Yeah,
No.
I saw it. So,
Okay.
but actually, I don't know this one where they come from. But right now, I know it's where they come from.
Uh,
So interesting.
so anyway, uh, so we tried to how this is fermentation, how fermented. Uh, Uruka, this is general knowledge. Uruka is one of the traditional shiokara food. Shiokara food is like using squid and ferment with high salt. Shiokara is very famous in Japan. And there are various uruka using different parts of ayus, such as gut, ovaries, and eggs. So many kind of uruka, but we focus on gut uruka. So if you have the freshwater rivers, then we can get ayu in Japan. So we have many urukas from many prefectures. So we got these uruka, and especially from Gifu City, we have some company, and I asked to get uruka produced uruka. So we got three kind of uruka from different prefecture. And in Gifu, we got uruka. One is early fermentation and middle fermentation and good, good late fermentation. And Uruka was believed as fermentation food, and we tried to isolate microbes like Narezushi, but we couldn't isolate microbes. And I told one student studied this kind of work, and told him, "You are very lazy. You should work hard." But indeed, that was wrong. And we tried to make Uruka in laboratory, and we checked. We couldn't isolate microbe, but the taste was Uruka in laboratory. So I, anyway, I tried because no one can eat. And I thought, oh, I told, oh, this is Uruka. I remember. And I am sorry that you are a lazy student. That is wrong. But anyway, so Uruka was believed fermentation food. But for us, this is not fermentation food. And we also analyzed microbes, microbiome using MGS. So, but you see, dominant genus are different among samples. So maybe Uruka is not microbial fermented food, but maybe enzymatically fermented food. So this is very difficult question. How to prove this? Student can understand how to prove. So probably no one have the answer. Only I have the answer. Have the answer. Anyway, I am studying about many kind of stress responses, and I am also studying pressure. Pressure is very convenient, high pressure. Okay, This is high pressure condition. We have the apparatus giving high pressure, about 100 megapascal. No. Indeed, more than that, we can. I think Yoren-san saw this apparatus. This.
Yeah, I saw it.
Yeah. So you see 100, 200 megapascal, maybe 200 megapascal we can increase using hydrostatic pressure, not gas pressure. Okay. We use gas pressure, but bang, it's very dangerous. So we use hydrostatic pressure. So using this, this is about 20 years ago. I wrote some paper and I knew that growth, microbial growth was limited until 100 megapascal. Viability is also limited around here. But metabolism can go 1 gigapascal or 400 megapascal. That means microbe cannot grow. And microbe maybe die. But enzymatic reaction can be carried out with this condition. So probably only pressure studying scientist knows this about this. So before we use high pressure, many scientists try to confirm enzymatic fermentation using antibiotics. But antibiotics is not complete to inhibit microbes. They just inhibit some microbes. But high pressure, we can inhibit the growth and we can decrease the viability. And no one knows we can maintain metabolism. So sublethal condition, no growth, no messenger RNA synthesis, no protein synthesis. But damaging. Oh, sorry. This is just for my papers, not this purpose. Anyway. So using 100 megapascal, 7 days and 30 degrees, this was enough, we thought. As we already made laboratory made Uruka using salt. So in this case, we don't have to use salt, but anyway, we use salt. I will explain later. Anyway, main point is this: 100 megapascal, 7 days and 30 degrees. In this condition, microbe cannot grow, indeed. Sometimes die. But enzyme can act and activation can be obtained. This is so indeed like, so if you put good inside and we give high pressure here, and we can keep this at 30 degrees. And we prepare homogeneate, only homogeneate, and we freeze it. And pressure and salt. And only pressure. Only salt. This is a general production procedure. And incubation only. No pressure, only incubation. You see big gases. And we also used Uruka from Shimane Prefecture and from Kochi Prefecture and Shiga Prefecture. And we have got early, middle, final fermented Uruka from Umaya is a company that produce Ingi food. And we can get his samples easy. So these samples we analyze using the ultra high performance liquid chromatography for amino acid, because amino acid is very important for taste. We try to see taste come from enzymatic or not. So using this machine, we characterize. This means this is a result. And so free amino acid in all 11 samples were measured using anyway, this chromatography. And this is the result. And this was analyzed by the principal component analysis for amino acid. You know, this is a statistic method. And we found, indeed, students didn't understand what is happening using this picture. But if you see this carefully, very easy to understand, okay? This is a homogenized sample. So this is just zero time, origin, start point. And salt alone is here, no movement. Pressure and salt, no movement. And umai early, no movement. But umai middle come to here. Umai final come to here. And Kochi prefecture here, Shiga prefecture here, and Shimane prefecture here. And pressure condition come here. So that means under pressure conditions, good our sample going to this Uruka. But using the salt, 1 week was not enough for producing Uruka. So if you use pressure, enzyme is active, but under salt condition, probably enzymatic activity is decreased. So we need several months for the fermentation. But under high pressure condition, we can produce Uruka in 1 week. So anyway, what we found is Uruka is not microbial fermentation, but enzymatic fermentation. Why aromatic fermentation? We don't need. Arabic fermentation is okay under pressure. So probably salt was added to prevent microorganisms growing, to kill micro. And for fermentation, we need several months or several years sometimes for Uruka producing. That is because of salt. Enzyme cannot act well. But under pressure, only 1 week is enough. So I told my producer that this is healthy Uruka. So you can sell. Do you have interest in high pressure treated Uruka? That is healthy because without salt. But Umaya said me, no, we don't need. So anyway, we could succeed. Uruka is not microbial fermentation, but enzymatic fermentation. So, and high pressure conditions can be used to distinguish between enzymatic fermentation and microbial fermentation. We have many Uruka-like food in Japan, and they say that is microbial fermentation is also essential. But probably that is not correct. We can isolate microbes from fermentation process, but that does not promise that that microbe contribute on fermentation. So they have to judge using high pressure conditions. So Yohani-san also in your lab, you can use high pressure. You can ask Yu-san,
Yeah,
see,
you sang
but
for salmon. I still remember you sang do it for salmon.
yeah. But carbon dioxide,
Yeah, carbon dioxide.
yeah, that
Mm.
is dangerous,
Yeah.
especially enzymatic fermentation. So anyway, we find new insight, especially Uruka is not microbial fermentation. So this company, Umaya, asked me, so Uruka is not fermentation. So we may say this is fermentation food. So I answered, so black tea or British tea is also fermented tea, we say. So enzymatic fermentation is also fermentation. Not microbial fermentation, but enzymatic fermentation is also fermentation. So you can say this is a fermentation. Uruka is a fermentation food. But they don't have interest in healthy Uruka. Okay. So next is Acetobacter that has been domesticated. You know, domesticated. Domesticated means like chicken. Chicken cannot fly, but they survive because we eat chicken. So they can, in that case, we say domesticated. And we focus on vinegar production. Rice vinegar brewing process are summarized. I asked Chappie to produce rice vinegar brewing process using English. And this is correct. Not exactly correct, but anyway, uh, using the sake brewing, Japanese rice wine is oxidized using acetobacter, and we get vinegar. That is rice vinegar. So later you can see these. And this koji and yeast and acetobacteria, and of course, this did not include, but lactic acid bacterium is also relating. Anyway, so as I told you about sake brewing process, and after that, we have no pasteurization and no maturation. After fermentation, that is directly go to vinegar production. Of course, after filtration, after removing some aggregate. And so very complicated process for um, rice vinegar. This is rice vinegar, normal rice vinegar, and black rice vinegar. And it is said this is good for health, but I don't know. Anyway, vinegar production process is similar. It's shown here. Raw material, carbohydrate, and saccharification, and alcohol fermentation. And without pasteurization, we go to acetic acid fermentation using acetic acid bacteria. And yes, so co-fermentation. In Japan, we use this kind of machine for host oxygen supply. We need a lot of oxygen for making vinegar. This is not traditional way. Traditional fermentation is not like this. But recently, we employ this kind of fermentators. This is European style, I think. And we need two kinds of tanks. One is for fermentation. One is for maintaining starter culture. This is maintained just aerobic, post-aerobic conditions because we couldn't isolate microbes from the tank for maintaining starter culture. They cannot make colony, indeed. And they have to maintain starter culture using this fermenter. So fermenting microbes cannot make colony or fermented by multiple microorganisms. We don't know, indeed. We have to know how it brewing. Anyway, but we found it, no, not we found. Anyway, we couldn't isolate. And this was also one student did, and he was not lazy. So I trust him. Oh no. So this is machines monitoring because this is a fermenter. We use fermenter. So many monitoring we can do using computer. And we can monitor many chemical component. So we just focus on rice vinegar ethanol. Ethanol, that means input amount rice vinegar ethanol means how much you put into the fermenter. And ethanol concentration, okay? We monitored, focus. This is ethanol content concentration. This becomes low. That means probably acetic acid produced. After that, they put ethanol again. So, and ethanol decreased. After decreasing, they put ethanol again, and we continue. And of course, we have to take sample, and that become vinegar. That is semi-continuous culture for using this kind of fermenter. And we monitored which kind of microbe, what kind of microbes are living. And from 1, 2, 3, and 4, 5, okay? 1, 2, 3, 4, 5. So until 3, there are so many microbes. But from the 4, it becomes only Komagatae, not only, but many others are, but dominant become Komagatae bacter. And from that, this is maintained. So probably Komagatae bacter produce acetic acid as a 4 and 5, 4 and 5. So after becoming dominant, ethanol drastically decreased. So there is some Komagatae bacter, but we don't know how it become dominant. Probably because of highly oxygen input become the make microbe killing, and this likes microbe like oxygen, and this become dominant After dominant, they produce acetic acid. So we try to what kind of species contribute on the fermentation because we cannot isolate microbes. We cannot make colony using the maintain starter culture. So for that, we used MySeq sequence and Minion sequence for hybrid system. So using hybrid assembly that incorporate both read sequence by MySeq and those sequence by Minion. The Zen sequence are reconstructed from both set of reads. So this is a short read sequencer, and this is a long read sequencer. We combine. And this is not isolated culture. So we need some depth, means many information we need. So we use this and this. Generally, MySeq is enough for isolated species, but this is complicated for only dominant. We have other species. After that, so this is the workflow of gene analysis using MySeq and Minion. Indeed, we just use some computer program, and we have this system in open some home pages. You can use that system that is already authorized. Anyway, using these kind of systems, we tried to get sequences. And after sequencing, we got two pins. Pins means gem. And bin one was estimated to have a complementation of completeness of just 6.41%. And bin 2 has 99.93%. And contamination rate was very low. So bin 2 is probably this. But bin 1 was maybe, I don't know where. I don't know where it come from. Anyway, we use this bin 2 as a genome. And using bin 2. And we try to identify which species. And we found Komagatai Europa else. This acetic acid bacterium were isolated from in Japan and in Korea and especially from Europe. As the company employs a fermenter similar to European system. So probably this strain was accumulated. I don't know how they accumulate, but anyway, probably this is a species. So, and yeah, indeed, these same species make colony. Only this cannot make colony, indeed. So this this was maintained in this fermenter for a long time. They lost like chicken. wing. They lost the wing. Well, they lost the colony forming capacity because they don't have to make. And the most important, new inside. What is new inside? Vinegar production. This is just Uchibori Brewing Company case. It's fermented by Komagata bacter Uruka else. And also, Europagenesis maintained in the many type culture collection, and they can make colonies. The vinegar fermenting Europagenesis has been lost to the colony forming ability. But we don't know. We couldn't find just analyzing genome, whole genome data. We have to analyze that. I have no laboratory, and someone may find the reason later. But if you have Japan and Indonesia is very famous for earthquake. If you have the accident of this Uchibori Brewing Company, they may get Komata bacter Uruka use from Europe, and they can start the fermentation again. Because if we have the big earthquake, we may lost this maintained tanks. So that is also suggestions from our soil. Okay, that's it for my lecture today.
Okay, thank you very much,
you.
Professor Hitoshi Iwahashi. For student, if you have any question, you can ask the question. But if you couldn't speak it in English, I can help you to translate to ask to Professor Iwahashi. Ada yang mau bertanya? Ya, Diva, you can ask anything from Professor Iwahashi.
Okay, thank you, Sensei, for your explanation. I have question about the bifidobacterium strain that was successfully isolated from Narezushi.
Yeah.
Yeah, the strain have salt and oxygen tolerance.
Yes.
But I'm curious that do you think this traits already exist before the strain entered Narezushi, or did they develop over time as it adapted to that environment? What do you think?
Where it come from, you mean?
I think my
Yeah,
question,
yeah,
this trait,
yeah.
salt and oxidant tolerance already exists before the strain enter that food, or did
No.
they develop over time
Yeah,
as it
yeah,
adapt to that environment?
yeah. You have a good question.
Thank you.
I don't know indeed. And indeed, when I submit this paper, reviewers asked similar questions. So you must be reviewer.
Maybe the reviewer is Diva.
Oh, yeah. And we have no answers about that. Maybe it is, but this is a Bifidobacterium cycloaerophyllum, probably from the first step. When entered to the brewery, this was a cycloaerophyllum and oxygen resistant, I think.
Mm.
Okay?
Okay, thank you for
Thank
your
you.
answer.
Thank you for question.
Any question from student? Any question from student? Majan?
Good.
So, so student, if you don't have a question, since I'm curious about this bifidobacterium because I
Yeah.
have a friends that from Egypt. Uh, how can I say that? The student from Egypt that doing experiments about bifidobacterium, and
Yeah.
she said to me that it's very difficult to maintain bifidobacterium.
Yeah.
And for Narezushi,
Yeah.
what is their function? What is their function in Narezushi, this bifidobacterium? Do you have any idea for that, Sensei?
Probably I have no idea.
Mm.
The
Okay.
maybe bifidobacterium produce acetic acid.
Acid. So,
So become the strange taste.
mm, so the taste is make it difference
Yeah.
maybe.
But
Yeah.
and every time we we can, I mean, just rare cases we found bifidobacterium. So bifidobacterium is not essential.
Oh, not essential in here.
Yeah,
Mm.
probably because no bifidobacterium here.
Mm.
Only in this case, only three cases.
Mm. Mm. Okay, okay.
Oh,
Yeah.
mm,
Had you ever tried to make the characteristic of this bifidobacterium, Sensei? Because this is the first report from fish, in fermented fish in
yeah.
Japan. So I think that it's better to, how can I say that, uh, analyze the characteristic of this bifidobacterium,
But
something like their biochemical or metabolic secondary that they have or something like that. Do you ever try to do that?
no,
Oh, okay, okay. Because it's not
because just
essential.
isolation and
It's
just
very
use
difficult.
a few characterizations.
Oh, okay, okay, okay.
And I all told the student that this maybe come from pig.
Why? I think, but this is for
Because we
tissue.
have this brewery was built about 150 years ago.
Yeah,
We
yeah.
have the World War,
Mm-hmm.
and that in that case, very complicated conditions in Japan.
Mm, yeah,
So,
yeah, yeah.
or,
Wow. This,
or owner
mm,
come here
mm.
and do something wrong.
Oh,
In
okay.
that case, came from human being.
Yeah, yeah, yeah.
So accidentally they live here.
Okay. So this one is from Kishu Naresushi. The beef
Yeah.
video only in Kishu Naresushi, right?
No,
Sensei?
not Kishonarezushi.
Yeah.
Only Yasuke is Yasuke Kishonarezushi.
How about Ayu Naresushi Sensei?
No,
They didn't. Oh, okay,
no.
okay.
Probably
Yeah.
only this Yasuke Narezushi.
Okay, okay.
Yeah, is the
Yeah, yeah.
case.
I think that if it's from Ayu Naresushi, they catch it by fish, right? So it's
Yeah,
maybe they have this beefy
yeah,
do
yeah,
it's from the fish stomach. Sorry,
yeah, yeah,
bird
yeah.
stomach. Yeah, yeah. Well,
That
it's
is
very
very
interesting.
reasonable indeed.
Yeah, yeah. Very interesting. Very interesting,
But
Sensei.
unfortunately, as I told you, we have only single
Mm,
from
yeah.
different sample.
Mm,
That means no diversity.
not diversity. Yeah,
So
yeah. Mm. So I think that for year you get this bifidobacterium, so it's very difficult to isolate them, right? Since
yeah, yeah.
it's very
Because
difficult.
lactic acid bacterium is contaminated.
Mm,
And
yeah,
lactic acid bacterium grow very fast.
yeah,
but bifidobacterium do not grow fast.
Mm.
We can see colony in 3 days.
Mm.
That
Yeah.
is lactic acid bacteria.
Yeah. Ah, okay.
We have you to until 6 days.
Okay.
After that, we can see small colony.
Oh,
From that, you
okay.
can get.
Okay.
Because students believe that once you see the colony, they get.
Mm.
But I told the student, you have to wait until more
Oh, yeah. Mm. Mm.
days. So,
Okay. So very interesting today.
yeah.
Today's topic is very interesting.
Yeah.
So for student, do you have any question? Do you have any question? Okay, I think that there is no question, so we can stop the lecture now. But I have a task for the student. For student get catch, I mean the student that choose food hygiene, bacteriology, and metabolic microorganism. You have a task for today lecture. So you should summarize for today lecture. And you can submit it 2 weeks later after this lecture. Untuk kalian yang mengambil mata kuliah higienitas makanan, bakteriologi, dan metabolik mikroorganisme, kita punya tugas, kalian harus ngerangkum kuliah hari ini, kemudian disubmit 2 minggu lagi. Okay, any question for the task? No question? Okay, Sensei, I think that today is enough.
Did you get PowerPoint?
Yeah, I get that's PowerPoint.
Okay.
Yeah.
So I will send this also.
Yeah, okay,
Yeah.
Sensei. Thank you very much, Sensei. Thank you for today.
So I have to
lecture.
prepare for the next week.
Thank you very much,
See you
Sensei.
next week.